Blend uniformity failure. This is the topic for the present video, and I welcome all viewers to the video. And let's start to understand about blend uniformity and blend uniformity failure. In the manufacturing of tablets and capsules and other solid formulations like granules, sachets, blend is prepared of the drug substance along with the excipients, and the blend is tested for uniformity and assay. Other parameters are also tested like LOD, water content, LOD or water content may be tested. Flow properties are tested, particle size distribution is tested, bulk density, tap density. These are the quality parameters of blend. And this blend is then used for the capsule filling, sachet filling, or tablet compression. Blend uniformity testing is a in-process testing before the tablet compression or capsule filling or sachet filling. It is also done for some of the products like dry syrups, powder for oral suspension, powder for oral syrup, and powder for oral administration. If blend uniformity fails, then it is understood that the process is not capable to consistently produce the drug product with quality parameters like uniformity of the dosage units or content uniformity and the assay. Why blend uniformity fails and what are the reasons behind it? How blend uniformity can be improved, that we will see in this video. Blend uniformity refers to the consistency of the drug blend in the batch. That means throughout the blend, the drug is equally distributed. Failure in BU testing indicates variability in blend composition, potentially affecting the drug product quality and patient safety. BU failure may lead to failure of the UOD, failure of the content uniformity and assay. And that's why immediate steps are required to investigate the cause and confirm if the failure is systemic or it is anomaly. That means the failure is only because of the analytical error, sampling error, or because of the process. So investigating the root cause is important, and if the first set of BU samples fails, then systematic evaluation is performed. Generally, if one set fails, then another set is tested. Generally, blend uniformity is performed in 1:3 to 3x sample size, 3x to 5x sample size, or the higher sample size to test whether the drug is equally distributed throughout the blend or not. And this sampling is done in triplicate samples, and first set is tested. If it fails, then investigation process starts to understand the root cause. Evaluate the blend parameters. Assess mixing time, speed and material addition sequence. Verify the blender design is suitable for the formulation or not. Then evaluate sampling procedure. So sampling thief issues, sampling rod issues, dye selection, sample collection method, sample size may introduce variability. Segregation during sampling, that means the sample is not representative of the entire blend. This problem may lead to sampling issues. Ensure sample sizes and collection locations are appropriate. So sampling issues are required to be investigated thoroughly. Then analyze the API and the excipients. Examine material properties. Check particle size distribution, density, flow properties of the blend, active material and the excipients. Investigate possible segregation or static charge issues that could affect uniformity. Also, hygroscopicity is required to be tested and understood to understand the root causes. Retest second set of samples. Retesting procedure is involving some steps like collect a new set of samples from different blender locations, and if you have duplicate sample available, then use that duplicate set of samples. Increase the sample size, minimum 10 samples for robust analysis. Nowadays, only 10 sample sampling is done in the pharmaceutical industry, and these samples are tested for the assay. Reanalyze blend uniformity to confirm if the initial failure was anomaly or systemic issue. So once you check the second set, you will understand the sampling problem or the process problem. Perform additional tests like content uniformity testing, evaluation of the content uniformity of the finished product like tablets, capsules as a secondary check. Stratified sampling. Conduct stratified sampling to assess variability within the batch, especially the critical areas. But as a general understanding and regulatory requirement for all the scale up and exhibit batches, we should always do stratified sampling. Stratified sampling along with the PU testing is done for some commercial validation batches as well. Blend uniformity acceptance criteria. There are certain acceptance criteria, and the most important criteria is blend uniformity acceptance sample within 90 to 110% of the label claim, and relative standard deviation, that is percent RSD, should be less than 5% unless justified for high dose, low dose products. So it is very important to understand the blend uniformity criteria, that assay of the blend should be within 90 to 11% of the label claim and RSD of not more than 5%. Another criteria is beta gamma criteria. Beta value measures variability within the sample means, and gamma value measures variability within the individual samples. But this beta gamma criteria is not that much utilized, and blend uniformity acceptance criteria of 5% RSD and assay within 90 to 11% is generalized. Content uniformity criteria. So once you complete the blend uniformity, then content uniformity should be done and tested. CU for the finished product should follow the USP chapter 905 for individual unit content, which should be within 85 to 115% of the label claim with RSD of less than 6%. Corrective actions after failure. If systemic issues are identified, then adjust the process parameters, modify the mixing time, speed, fill levels, and sequence. You can also check the actual occupancies. Improve sampling techniques. Use validated sampling methods to minimize disturbance. Ensure consistent sampling from the critical locations. Implement process analytical technology, that is PAT. Use tools like near IR spectroscopy for the real-time monitoring. These are some of the important aspects. Investigating and addressing blend uniformity failures require structured approach. Identify the root cause by evaluating the process, materials, and samplings. Retest to confirm whether the issue is anomaly. Implement corrective actions such as adjusting parameters, improving sampling, and utilizing the real-time monitoring. So these are the best practice. So this is regarding the blend uniformity failure and blend uniformity improvement techniques. I hope this video will help you to understand the blend uniformity criteria, blend uniformity failure, and the best practices to avoid the failures in PU. As per my experience, if you find variation in the blend uniformity samples, then you should go for compact sampling for BU sampling rather than the powder sampling. Also, always try to sample the 3x to 5x sample size so that you will have good amount of blend or powder quantity to minimize the variation and improve the analysis. Thank you for watching the video, and let's meet soon with such kind of informative video.